Welcome to the Sand Science Experience, your source for the most current and impactful research and preprints in science. Please take a moment to like this video and subscribe to our channel. It truly helps support the Sand Science Experience. Okay, let's unpack this. Today we're diving deep into a really fascinating cellular puzzle. It's all about how tweaking the powerhouses of our cells, the mitochondria, can surprisingly impact lifespan across different species. Our sources kind of take us on a journey, from simple worms right up to mammals.
Yeah, and what's really intriguing here is getting into the core mechanism. So every cell has these tiny engines, mitochondria, right? Mostly known for making ATP, the cell's energy. But here's the twist. They have their own protein factories, their own ribosomes, and these make essential proteins for energy production itself, like key parts of complex 1 and complex 4.
Okay.
Now, when this mitochondrial protein making gets subtly perturbed, let's say it can trigger a kind of cellular alarm system. We call it the mitochondrial unfolded protein response, or UPRMT.
UPRMT, right.
And the really surprising part, sometimes this mild stress actually signals the cell like, hey, let's clean house.
Yeah.
And this leads to changes that can, extend life. It's like a little cellular tune-up.
A cellular tune-up. I like that.
Mm-hmm.
So if a little tune-up is good, does that work for everything? Let's start with the worms. C. elegans. What does the research show there? Seems pretty remarkable.
Oh, absolutely. In worms, a modest inhibition, just dialing it back a bit, robustly extends their lifespan. Researchers found a few ways to do this. For instance, a small genetic tweak in a mitochondrial ribosome gene.
Which gene was that?
It was MRPL2. This reduced translation turned on that UPRMT alarm, interestingly, in a vitamin B12-dependent way, and the worms lived longer.
Huh. Vitamin B12, that's specific. What else?
Well, another experiment involved knocking down TARS1. That's a gene for tRNA synthetases, sort of cellular translators for protein building. Doing that boosted lifespan significantly, sometimes, 40 to 60% increase.
Wow, 40, 60%, that's huge.
It is huge. Again, it activated UPRMT, but it wasn't all good news. There were some developmental delays, and the timing was absolutely key. These things work during larval development, not in the adult worms.
Ah. Okay, so that's a crucial detail, a developmental window. And it seems like these mild disruptions cascade into other good things. The sources mention combining things.
Exactly. They combine knocking down a ribosomal protein, MRPS5, with disrupting mitochondrial fusion. That's how mitochondria connect.
Right, using EAT3 knockdown.
That's the one. And doing both together. Even longer lifespan. Plus, it ramped up the creation of lysosomes, the cell's cleanup crew, via this protein HLH30. So yeah, it looks like enhanced cellular recycling, autophagy.
So the cell is literally cleaning itself up better.
Precisely. But it really highlights this delicate balance. It's not just about less is more. Get this. Making mitochondrial translation hyper-accurate, too perfect, can actually reduce lifespan, even though UPRMT gets activated.
Seriously, too perfect is bad.
Seems so. And moderately error-prone translation didn't do much either. It's like you need this Goldilocks zone. Mm-hmm. Both the rate and the accuracy have to be just right.
Okay, that makes sense for worms, a Goldilocks zone.
Yeah.
But does this whole idea hold up as things get more complex? What about fruit flies, Drosophila?
Flies? Oh, well, the story there gets a bit more nuanced, especially when you factor in diet. Under dietary restriction, DR flies actually do the opposite in some ways. They increase the production of certain nuclear-encoded mitochondrial proteins.
Like complex 1 and 4 components.
Exactly, via a pathway called D4EBP, and this boosts their respiratory function and extends lifespan. So, in that specific context, DR in flies enhancing certain mitochondrial translation is good.
Huh. So context is everything.
It really is. And conversely, if you mess up key regulatory factors for mitochondrial mRNAs in flies, like DMLRP-TRC2, lifespan shortens because their energy production tanks. Not so simple.
Definitely not simple. Okay, so worms show benefits from slight inhibition. Flies are complex and diet dependent. What about mammals?
Mice. Yeah, moving to mice, the link gets even weaker, or at least less straightforward. For instance, one study looked at mice with about 50% less of a mitochondrial ribosomal protein, MSRPL54. They saw basically little to no significant lifespan extension, or metabolic health benefits for that matter.
And did the UPRMT alarm go off? Like in the worms?
That's the key thing, no. The UPRMT wasn't activated in those mice. It really suggests that mammals might just have a much higher threshold. You might need to inhibit mitochondrial translation a lot more to trigger those potentially beneficial stress responses.
A higher threshold. Okay, so our systems might be, more buffered against this kind of stress?
Could be. More robust, perhaps. Or maybe the signaling networks are just wired differently. It raises big questions.
It certainly does. Which leads to the next step. What about zebrafish? Or, us, humans?
Well, for zebrafish, there's just not enough direct experimental evidence on this specific angle right now. It's a gap. And for humans, look, we absolutely know mitochondrial problems are linked to aging and disease. The basic machinery is there, it's conserved. But is there direct experimental evidence that tweaking mitochondrial translation extends human lifespan? No, definitely not. There are huge ethical and technical hurdles, obviously. It's mostly theoretical discussion at this point.
Right, makes sense. So summing up this deep dive then.
Yeah.
It feels less like a simple switch and more like a really complex balancing act.
That's a perfect way to put it.
We see moderate disruption helping in worms, maybe flies under certain conditions, but it gets tricky fast. You've got this interplay between mitochondria and the rest of the cell, the autophagy connection, like with HLH30 in worms, diet effects.
And that need for an optimal range. Not just the rate of making proteins, but the fidelity, the accuracy too. It all needs to be balanced for healthy aging, for proteostasis.
So to wrap it up, perturbing mitochondrial translation can trigger adaptive responses and extend life in simpler organisms, but it's way more complex in mammals. Mice show that higher threshold.
Indeed. So while manipulating mitochondrial translation seems promising in those simpler models, actually translating that to vertebrates, let alone humans, well, that's still a major challenge, an open frontier. It really makes you think, though, doesn't it? Just how finely tuned our cellular systems have to be for longevity. What stands out to you about that intricacy?
Absolutely fascinating stuff to ponder. And remember, everyone, please like this video and subscribe to our channel for more deep dives into the latest science. Thank you for joining us on the Sand Science Experience.